Hello, my name is Miles Davis, and I will be discussing the technique called immunofluorescence. So, immunofluorescence was first developed by Albert Kuhns, who was a physician, pathologist, and immunologist. And this specific technique was developed in 1950. So, what can immunofluorescence be used for? So, first, it can be used for visualizing particular proteins and antigens. It can be used to identify particular cells, track protein movement, and determine co-localization. And so if you look on the right here, you can see an image of an interferon and neurofilament being immunostained with red and green. You can see further to the right and on panel B that both of these happen to be co-localizing in the same region of the cell. So some materials that are needed for this particular technique are a fixative. One specific fixative that can be used is PBS. Permeabilization solution. An example of this is Trident X100. A blocking solution, which can be used that can be used is BSA, a primary antibody specific to a certain protein, a secondary antibody, which will be used to bind to the primary antibody. The secondary antibody will also contain a fluorophore and either a epifluorescence or confocal microscope to view the particular sample. All right, so in order to do this, you must fix the sample. And so what this means is you want to make sure that nothing in the cell is moving and it's stayed in a fixed spot. And so this can be done by incubating the sample for 10 minutes at room temperature in a 4% formalin solution. And as stated before, this is going to leave the cells in a fixed position along with their proteins and organelles. Here we see that you must create a permeable membrane of the cell. We do this by incubating it with a detergent, and a lot of the time the permeabilization solution must be specific to a particular antibody because, as we'll see later on, the antibody must be able to enter the cells once being made permeable. Here we see that the next step is blocking. So what this does is the blocking solution or the blocking agent is going to bind to the non-target antigen. And what this does is that it's going to prevent the primary antibodies from binding to the incorrect antigen. And so this is done by incubating the sample in a serum of the host in which the secondary antibody was made. And as stated before, a common blocking agent is BSA. We then move on to the primary and the primary antibody incubation. And so here what we're doing is we're adding the primary antibody to the sample, and it's able to go into the cell because it is not permeable to the specific antigen. This antigen can be, or any antigen can be used and produced by host organisms. And so what they do is they will inject the host organism with the specific protein that they're looking for it to bind to. And after a certain period of time, these host organisms will develop antibodies to the specific protein. and these antibodies will then be extracted from the organisms. And so going forward, once the sample is incubated with the primary antibody, it is going to be washed 3 times for 5 minutes in a washing solution. So the next step is going to be creating a secondary antibody. And so, very similar to the primary antibody, we'll be using a host organism to create these. But the only difference is that a fluorophore is going to be attached to the secondary antibody. And so what happens is once the sample is incubated with the secondary antibody, it will bind to the primary antibody, and this should show us specifically where this protein is, it being in this particular part of the cell. It is very important that this reaction takes place in the dark so that the fluorophores do not become excited. Once incubated, the sample must be washed 3 times for approximately 5 minutes in a washing solution. And so after this, they must be counterstained and mounted. So the mounting a mounting medium is added to the specimen. And so what this does is that it protects it from any damage or degradation in this process. And counterstain is added, commonly DAPI, which can be used as contrast so that you're easily able to see the fluorescence in the sample. And once this is done, you can then view this sample in either a epifluorescence microscope or a confocal microscope. And in this specific example, as you can see to the right, it is a neuron that has been immunologically stained. And so my specific example that I found was a study called the use of 4-color immunofluorescence techniques to identify mesenchymal stem cells. And so here, this study was conducted by Schieker and colleagues in the Department of Surgery at Ludwig Maximilians University in the year 2004. And so at the time, there was no way to differentiate HMSC stem cells on the single cellular level. And the current ways that they did have to identify these stem cells, it would cause the stem cells to lose their stem cell characteristics and their pluripotency. And so what they wanted to do was generate an image using immunofluorescence to compare HMSC cells to other cells to be able to differentiate them. And so here they used 4 different types of proteins: collagen 1, collagen 4, fibronectin, and CD44. These 4 proteins were all targeted by primary antibodies. They used these specific ones, collagen 1 and CD4, because they were early markers that this was the cell that was being looked at is a HMSC cell. And they used collagen 4 and fibronectin, one because collagen 4 is only expressed in HMSC, and fibronectin is strongly expressed in HMSC. And so here you can see on the right of each of the proteins, there is a different fluorescence that is used. Collagen 1 uses FITC, which shows green. Collagen 4 uses Alexa 546, which shows orangish or yellow. Fibronectin is Texas red, which shows a red color, and CD44 shows AMCA, which is a blue color. Here, this table pretty much shows what specific host they used for each of the antibodies. And so here you can see fibronectin was made with mouse antibodies. Collagen 1 was used, the host organism was a rabbit. CD44, the host organism was a rat. And in collagen 4, the host organism was a goat. And they did something very similar with the secondary antibodies, but they made them specific to each of the primary antibodies to make sure that they can attach to them. And so here what we can see is A and B being a virtual and spectral image accordingly of each of the protein types being expressed. You can see that the next images, C through H, are the expression of each of the 4 different types of proteins. Collagen 1 is shown in the green spectra being E and H. Collagen 4 in the yellow spectra which is going to be slide F. Fibronectin, which is shown in Texas red, in the red spectra, D and G. And CD44 as the blue spectra shown in C and K. And as stated before, A and B is the combined image of all of these, showing the expression of all 4 of the proteins. So overall, the creation of this image was successful. And what they wanted to do was compare this picture to, or this image to other cells, potentially other cells, as well as showing the expression of HMSC specific proteins to prove that the cell that's being viewed is this particular type of stem cells. And they wanted to use this technique to be able to identify other types of cells using specific proteins that may be specific to those certain cells. And these are my references. Thank you.